Hey everyone, today we're going to be looking at DNA and protein synthesis, but this video specifically is going to be looking at DNA and RNA structure, similar to what we discussed last year, with just a few more details, a little more depth in certain areas. Some of the things that you should already remember is that DNA and RNA are both polymers, so they're made up of smaller units, and both DNA and RNA are made up of those small units, which are called nucleotides. So we can also call DNA and RNA polynucleotides because they're made up of a bunch of smaller subunits called nucleotides. So when we look at the monomer unit of DNA and RNA, those monomers, those small building blocks that we link together, remember, are called nucleotides. And there's a slight difference, once again, between the DNA nucleotides and the RNA nucleotides. If we look at DNA structure, we already remember that it's a double helix structure. For some reason, I'm not sure why, people just relate acids together. Nucleic acids have no structural relationship to amino acids. They're nowhere near the same. Just because they both have the word acid in it has no meaning that they're actually made of each other or directly deal with each other. So amino acids, remember, make proteins. Nucleic acids are a completely different building block made out of nucleotides. And then specifically for a DNA nucleotide, remember there's three. This entire picture right here is the nucleotide. Remember, there's three individual parts of a nucleotide. You have the deoxyribose sugar, also a type of pentose sugar. You see it's a pentagon shape, a 5-carbon sugar, 1, 2, 3, 4, 5. You have the phosphate group, and then you have the nitrogenous base, the adenine, cytosine, guanine, or thymine in DNA. And all three of these things together, it's what creates that DNA nucleotide or that deoxyribonucleotide. Comparing that to RNA, remember RNA is single helix. RNA does not have thymine in it. RNA has a very similar structure, very similar base called uracil. You have your three main types that we'll deal with: tRNA, mRNA, and rRNA. And the second big difference between the RNA and DNA is, remember, DNA has deoxyribose, that's why it's deoxyribonucleic acid, while RNA has the ribose sugar, which is also still a pentose sugar. It's still a pentagon shape, but has an oxygen that deoxyribose does not. But it still has nucleotides made of ribose, phosphate, and then one of the 4 nitrogen bases, A, U, C, or G. Why is DNA and RNA so important? As we know, DNA is really that directions on how to actually create a protein. It also has the ability to direct its own replication, so it can, through base pairing, help itself replicate. It can't do it by itself. RNA can self-replicate, but DNA has the instructions on how to actually create itself through that base pairing mechanism. Also, our DNA has the code, as we said, to actually create that protein, and that DNA is really that hereditary molecule that's passed from parent to offspring. Looking at the different types of RNA that we deal with, you have messenger RNA, which is single-strand RNA, as we'll deal with in more detail, but hopefully remember it's created during the transcription step of protein synthesis, and it is also created using base pairing rules, so A to U and then C to G in terms of looking at the base pairing. So you have AAA here on the DNA template strand, which means you must have UUU when you're creating that mRNA strand. So that mRNA has to be complementary to the template DNA strand. Comparing that to ribosomal RNA, rRNA, which is part of the structural components of the ribosome, of the either 70S ribosome or 80S ribosome, depending on if it's a prokaryote or a eukaryote. As we also already know, the ribosomes are formed in the nucleolus, and that's where the rRNA is actually created. That's why it's that dark spot, is there's so much transcription happening. There's so much rRNA being produced constantly. That's actually why it's that darker shade, because there's so much nucleic acids in that area to actually stain, so it appears darker. And once again, this forms the ribosomes also with some protein components that actually creates that structure right here, this cartoon structure that's actually going to translate that mRNA into that amino acid sequence. And then the last type of RNA that we deal with is tRNA transfer RNA. It's a single strand, so it's one strand, but it's folded it actually base pairs with itself and creates this shape. In real life, and this is a two-dimensional shape. In real life, these two sides actually fold back a little bit, so they create like little wings almost on the tRNA molecule. But you can see in the unfolded shape, the flat shape, it almost looks like a T shape. Has this clover leaf shape. You can see the loops at these three loops. One side of it is the acceptor stem, which is where the amino acid is going to bind, whatever specific amino acid for that specific tRNA molecule. Remember, each tRNA molecule can only carry one specific amino acid. And then the other side, you have the anticodon loop, which is where the three anticodon bases are going to exist. And where those anticodons are what's going to bind to the codon on the mRNA and actually deliver that amino acid in the correct spot of that actual protein structure. So every one tRNA molecule can only carry one specific amino acid because there's only one specific anticodon on that tRNA molecule. If we look at the flow of information for creating a protein, remember that the translation step is done by the ribosome. So you start out with the DNA, which codes the information on how to create that protein, is the actual order or knows the order of the amino acids. You have transcription, which is going to create that mRNA in the nucleus. The mRNA will then leave the nucleus and go out either to the cytoplasm or to the rough endoplasmic reticulum. That mRNA is then going to get read during translation by the ribosome. tRNA molecules will come in based on that anticodon-codon base pairing. They'll drop off the correct amino acid in the correct spot. Those amino acids will be then linked together through condensation reactions. And then it's going to create this growing polypeptide, which eventually is going to fold into a functional protein. As we already mentioned, this nucleotide is made up of three different units. You have the base over here. You have the pentose sugar, depending on if it's a RNA molecule, have ribose. If it's a DNA molecule, have deoxyribose. And then you have the phosphate group over here, which you can see is already connected to the next nucleotide in the phosphate group. How do you actually form a DNA and RNA structure? The actual chain of nucleotides. Just similar to building any other type of molecule in this class, basically. It's a condensation reaction where you can see you have the phosphate group, you have the hydroxyl group on the phosphate group, interacts with the hydroxyl group on the pentose sugar. The water is removed and it creates this bond right here. This entire thing is called a phosphodiester bond. So the ester bond is this bond between the oxygen and the phosphate, similar to the ester bond in fats. So you have this is the ester bond. This entire thing is the ester bond. And so you have an ester bond here, and then you have an ester bond here. So that's where the phosphodi, as in two ester bonds. So one ester bond here, second ester bond here, and this phosphodiester bond is what's maintaining the phosphate sugar backbone. And this is a very strong, it's a covalent bond, so it's very strong, similar to all the other bonds, similar to the peptide bonds in proteins and the ester bonds in fats. So it's an extremely strong bond, so it's not going to break easily. So it has that structural component, maintaining the DNA structure. And we can see here is an entire nucleotide unit, and then the phosphate sugar backbone is on the outside of it, and then you have the bases that actually stick out to the inside, or in this case this is RNA, so they just stick out, and then DNA, they stick out to the inside, and then you have the complementary strand, once again base pairing ATCG, and we can see the hydrogen bonds are what maintain the double strand nature of DNA. So the phosphodiester bonds, the covalent bonds, maintain the sugar phosphate backbone, and then the hydrogen bonds maintain the two strands being actually stuck together. We also already mentioned this last year, the slides from last year actually, that DNA is anti-parallel, so it's like a two-lane highway where you have one lane going down, one lane going up. So you have a 5' end and a 3' end. 5' end is the one with the phosphate group, 3' end is the one with the hydroxyl group, the OH group, and they just run in opposite directions. So you have this 5' to 3', so they're just in opposite directions. That's pretty much what this is saying. Unfortunately, last year this was not something you need to know, but this year it is, that great, you know A, T, C, and G, you know your base pairings. But unfortunately, there's a little more detail. You do need to be able to recognize more of the actual physical, real structure of the nitrogen bases. And so we have two different types. We have pyrimidines and purines. Pyrimidines are single nitrogen rings, and they are examples of pyrimidines are cytosine, thymine, and uracil. So there's only one ring shape for that nitrogen base. Comparing that to purines, which have this double ring shape, this double nitrogen ring, which are adenine and guanine. And you can see that in terms of pyrimidines and purines, every one pyrimidine bonds to one purine. So you can see the cytosine is a pyrimidine. It will bind to the purine guanine. Thymine is a pyrimidine, which will bind to the purine adenine. And one of the things, the way I actually remember what is pyrimidine's a longer word, and it's actually a much simpler diagram. Purine's a much simpler word, but it's a larger diagram. And that's how I memorize which ones are pyrimidines, which ones are purines in terms of the ring structure. If you have a different way, that's great, but I just remember the bigger the word, the less actual structure there is. And how do I remember which ones are purines, which one are pyrimidines? I don't remember the pyrimidines, but I remember the purines because they're ag, A and G. I can remember ag is purine. So ag is purine. That means the other three bases have to be pyrimidines. And so you can see in more detail the A, T, C, G, and U. So we have adenine and guanine. You can see are very, very similar. Just slight differences. This adenine has an NH2 group, while guanine has a carbonyl group. You can see he has a double bond here, comparing to only a single bond here and an extra hydrogen. Similar idea with cytosine and thymine. Very similar structures, NH2 versus an O. Double bond here versus wait, sorry, not double. Sorry, it's the same. Here has just an H, and here has an entire methane group that's sticking to it. And you can see uracil is also very, very similar to thymine. The only real difference is the lack of the methane group that's sticking off here. And please, when you talk about sugars in the terms of the DNA and RNA structure, you cannot just call things sugars. You have to actually say it's a deoxyribose sugar or a ribose sugar. If you just say sugar, that's not like specific enough for the AICE exam. So please make sure you use the actual word deoxyribose or ribose, depending on the actual nucleic acid we're dealing with. And then putting it all together, if we just have a nitrogen base and a deoxyribose, we call that a nucleoside right here. If we add that phosphate group, we call that a nucleotide. And then if we add more phosphates to it, we can have a nucleoside diphosphate like ADP. And if we add another one, we can have a nucleoside triphosphate, ATP. So we can just add more phosphates. So this is a monophosphate, it's called just a nucleotide. Then you have a diphosphate and then a triphosphate like ADP and ATP. In terms of Chargaff's rules, we know the complementary base pairing A, T, C, and G. One thing that you actually do have to memorize this year is how many hydrogen bonds actually form between the A, T, C's, and G's. So C and G actually form 3 hydrogen bonds, so it's a much stronger bond because there's 3 of them, while A and T actually only form 2 bonds. So adenine and thymine will only form 2 bonds, while cytosine and guanine will actually form 3 hydrogen bonds. So C's and G's are much harder to rip apart than A's and T's. And we'll discuss that in a little detail in terms of transcription and where transcription actually starts and why it actually starts there. And what's so important about nucleotides? So remember, it's that order of these nucleotides, these order of these bases that actually make you. So your nucleotide sequence is different than another person's nucleotide sequence unless you have an identical twin. And it's the code, that actual order of those the order of the nucleotides, which can change the order of the amino acids, which can change your protein, which will then can change how you actually are, your phenotype. So it's these nucleotide sequences that make up your genes, and then all your genes make up what's called the genome, which is your entire DNA sequence inside your cells. And basically, you already said this, that the entire point of nucleotides is that you read them off in groups of 3. not the entire point, but for the main point of what we're dealing with, is that you read them off in groups of 3 in each gene, and then that codes for a specific amino acid, which then creates a specific protein, which then determines a specific protein and does a specific function.